Yeah. Our first speaker is Takashi Okada from Kyoto University. Please start, Okada-san.
Yes, thank you very much. My name is Takashi Okada from Kyoto University, and it's honor to speak here. And I'm excited to tell you about our studies on chemical reaction networks. And today, I'd like to present on some methods of analyzing steady-state properties of reaction networks based on network topology. Okay. And so let me start. So I'm studying chemical reaction networks mainly based on biological motivations. Specifically, we want to understand how cells regulate their states or phenotypes through parameter control. Here, parameters can be whatever, but in the context of metabolic networks, enzyme expression levels can be regarded as parameters. And in the context of signal transduction network, shown on the right, you can consider external stimuli as control parameters. And a major theoretical challenge is that for many reactions inside cells, we don't have a precise knowledge of reaction rate functions or parameter values. So this raises a question. Can we understand the behaviors of these systems from network structure? And it turns out that many properties of chemical reaction networks are determined by their network structure alone. Okay, so, and I think most of you are familiar with the basics of chemical reaction networks, but to introduce some notation, I quickly review the dynamical system of a chemical reaction network in this slide. So today I'm focusing on the deterministic dynamics. And so deterministically, the dynamics is described by ordinary differential equations shown here. So here, Xm represents the concentration of molecule M, and Ri is the rate function of reaction I. And the rate function, of course, depends on concentrations, and also depends on some parameters, Ki. So for example, in the context of metabolic network, each reaction is catalyzed by enzymes. So Ki is represents the enzyme activity or enzyme amount that catalyzes the reaction I. Okay, and finally, this matrix S is so-called the stoichiometric matrix, and that represents how many molecules are consumed and produced at each reaction step. So for example, in a toy network on the right, here there are 3 chemical species, ABC, and there are 5 reactions. And the dynamics are described by these 3 equations. And for example, xb dot is given by R2 minus R3 because B is produced by the second reaction and consumed by the third reaction. So we have R2 minus R3. And these equations can be written compactly in matrix notation like this. And this matrix is a stoichiometry matrix. And finally, about reaction rate functions, people often assume specific kinetics like mass action kinetics or Michaelis-Menten kinetics. But as I mentioned earlier, we don't have a precise knowledge about what kinetics are realized in living cells. So in our approach, we don't assume these specific functions, but we only assume that each reaction rate function depends on its substrate concentrations. So for example, for example here, I included XA in the argument of the reaction 2, because A is the substrate of the reaction 2. Okay, so the rate of the second reaction 2 should depend on the concentration of chemical A. Okay, and so with this very general setting, let's discuss steady state properties. Specifically, I'd like to first discuss the steady state sensitivity. This is basically, this is actually done originally by Bernard Fedora and Mochizuki-san. Okay, so specifically, I here I like to discuss the steady state sensitivity. So, so here x bar m denotes the concentration of chemical M, and then this is this depends on parameters in general. So here, what I'm asking here is that which parameter this concentration depends on. And we can systematically answer this question by using linear algebraic computation. And I'd like to first explain the result. To compute the sensitivity, we first construct a matrix A shown here. So here A consists of two parts: the reaction rate derivatives and reaction rate. And the kernel vectors of the stoichiometric matrix S. And the sensitivity is given by the inverse of this matrix A. And the proof is actually very simple. So let me explain this, the proof. So we start from the steady state conditions. So zero, we can set the time derivative to 0. And this equation means that the reaction rate vector must be in the kernel space of S. So we can expand the reaction rate vector in terms of a basis of the kernel space, C. And here mu alpha denotes the coefficient. And differentiating this equation with respect to parameter, for example, Kj, gives this the third line. And this equation can be rewritten like this using the matrix A. So A times the sensitivity vector is proportional to the unit vector. So the sensitivity, this vector can be obtained by, is given by the inverse of, minus times the inverse of matrix A. Okay, I think this is, so the argument very easy. Yeah, so, sorry. And the important point of this method is that the matrix A is typically a sparse matrix that reflects the underlying network structure. So, whether each element of each matrix element of A is 0 or non-zero can be determined from the structure information. So we can also determine whether the sensitivity is 0 or non-zero from the structure information. And I like to illustrate this point in the previous example. So for this network, the matrix A is given by like this. So the matrix A consists of two parts: reaction rate derivatives and the kernel vectors. And for example, R2A appears here because reaction 2 depends on the concentration of A. Okay, so we have non-zero elements on the left, and these constants, the vectors are is a basis of the kernel space. So in this case, we have two basis vectors. And yes, sorry, here, I forgot to explain this. RIM denotes the reaction rate derivatives. And usually, reaction rate functions are increasing functions of concentrations. So usually we can assume that RIM, these small r's are positive. So we can determine the sensitivity as shown, the signs of the sensitivity as shown here. Okay. And I'm showing the this result below. So for example, so here each triangle denotes a particular perturbation to a parameter. So for example, here this means that the perturbation to the reaction 4 decreases the concentration of A and B. And a bit surprisingly, the concentration of C is not affected, even though it is C is downward of the perturbed part. It's similar to for the perturbation to 3. So perturbation to reaction 3 affects only B, the upward of the perturbed part, and downward is not responded. And but anyway, we can determine which part of the network exhibit non-zero responses from this matrix computation. And we found a general theorem that determines which part of the network will exhibit non-zero responses to a given perturbation. And here's a theorem. Suppose that a subnetwork gamma satisfies the following two conditions. First, the reactions of gamma includes all reactions whose rate functions depends on chemical concentrations inside gamma. So I mean, so that is so suppose that this red species is a member of gamma, then these two reactions, these two emanating reactions, yes, so the reaction rates of these two reactions should depend on the concentration of this red chemical species. So we need to include these two reactions as member of gamma. Okay, this is the first condition. And second, we calculate a kind of index, which is defined by as the number of chemicals inside gamma minus the number of reactions inside gamma plus the dimension of the kernel space inside gamma. And roughly speaking, the last term counts the number of cycles inside gamma. So yeah, so this formula is very analogous to the Euler characteristic in graph theory. And let's suppose that this index is equal to 0. And we call such a subnetwork a buffering structure because for such a subnetwork gamma, we can show that any parameters inside gamma do not affect the outside concentrations of gamma or outside reaction rates of gamma. So in some sense, such a subnetwork can confine the effect of the perturbation inside it. And these are, here I'm showing some examples. So here the part, the subnetworks enclosed by orange lines are buffering structures. For example, in the second case, we have two chemical species, A, B. So we have two here, and we have three reaction edges, so 2 minus 3. And we have one cycle, one loop here. So the loop consisting of reaction 2, 3, 4. Okay, so we have one loop. So 2 minus 3 plus 1. So the index is 0. So this is a buffering structure. And consistently with this theorem, the perturbation of perturbation to reaction 4 does not affect the outside, the concentration of C. Okay, so in this way, we can we can understand which part of the network is influenced by a given perturbation based on some structure conditions. And the proof is actually very easy, but I don't go into the details. Yeah, but basically, these two conditions imply that the matrix A has a block triangular shape, as shown here. And in particular, the second condition implies that these two blocks are square blocks. And for such a matrix, we can easily compute the inverse of the A. And in particular, yeah, zero matrix again appears in the inverse, and this zero matrix on the lower left corner implies zero sensitivity outside the buffering structure. Okay, and yeah, so I'd like to show some more examples in a slightly larger network. Here, this is central carbon metabolism of E. coli bacteria, and here the glucose is taken from the outside, and it is decomposed into smaller chemical species. And there are many buffering structures in this network. For example, this subnetwork is a buffering structure. This yellow part is also a buffering structure. And this yellow part plus red part is another buffering structure. And there are 17 buffering structures in total. And yeah, so and we can consider the inclusion relation among these 17 buffering structures. And this diagram shows the inclusion relation between the 17 buffering structures. And but this diagram looks a bit complicated, so let me explain this this pattern in a simple way. So firstly, TCAC, this blue pathway, is a buffering structure, and the yellow part, PPP, is also a buffering structure. And finally, the remaining part, the glycolysis, glycolysis is not a buffering structure, but combining the glycolysis with PPP and TCAC gives a buffering structure. In other words, the whole network is a buffering structure. And the inclusion relation between buffering structures is important because it implies a hierarchy in enzyme regulations. So please recall the property of buffering structures. So according to the property of buffering structure, when PPP, so because PPP is a buffering structure, enzymes catalyzed in PPP affect only metabolites inside the PPP. So the enzymes catalyzing this yellow pathway affects only these three metabolite concentrations. Similarly, because TCSC is a buffering structure, enzymes catalyzing these reactions, these blue reactions, affect only chemical species metabolite concentrations in the blue pathway. But the glycolysis is not a buffering structure. Combining glycolysis with PPP and TCAC gives a buffering structure. So enzymes catalyzing glycolysis affects not only metabolites in glycolysis, but also those in PPP and those in TCAC. So in this way, so as long as the network information is correct by examining buffering structures and examining how these buffering structures form the inclusion relationship, we can obtain qualitative understanding about how the system is regulated by enzymes. Okay. Okay. So and finally, I like to briefly comment on some properties of buffering structures. Firstly, buffering structures are closed under intersection and union. So if you have multiple buffering structures, you can consider their unions or their intersections, and they are still buffering structures. And second, so far, I implicitly assume that the system has no conserved quantities. But in general, a chemical reaction network may contain conserved quantities. For example, in this simple net example, the sum of the concentrations of A and B is obviously conserved. For a net and for such a network, we need to modify the index slightly, like this. So the first three terms are the same as the case without conserved quantities, but when the system has conserved quantities, we have the additional term shown here. And this the last term counts the number of conserved quantities inside the buffering structure. And with this modification, we can show the same thing even when the system has conserved quantities. And finally, the inverse of the theorem is also true. I mean, so if some chemical species are, so if some chemical species are not affected by parameter perturbation, then we can show that there must be a corresponding buffering structure that explains the zero sensitivity. Okay. And the last property in the previous slide is indicates practical method for finding buffering structures. So far, I haven't explained how to find all buffering structures for a given network, and we can practically find all buffering structures by computing steady-state sensitivities. So this comes from the last property in the previous slide. So specifically, for example, below are the sensitivities obtained from the matrix computation. And these zero sensitivities, the non-zero patterns should be explained from buffering structures. And in fact, from these, there are corresponding buffering structures for each non-zero pattern. Okay. Okay. So okay. So in the remaining time, I'd like to talk about another topic, network reduction based on buffering structures. Yeah. Okay. So to clarify the problem, let's suppose that there is a network, and that contains several buffering structures. Here, buffering structures are shown by a green boxes, two buffering structures. And let's suppose that you are interested in the steady-state properties of the yellow part, not the green, and you are not interested in the green parts. Then you may expect that for this purpose, we can ignore the green parts because as I said, parameters inside the green part does not affect the outside. So if we are interested in the yellow part, or we naively expect that we can ignore the green parts like this. And in fact, this expectation is almost correct. But there is a subtlety. If you simply remove the green parts, ignore green parts, then the resulting system will not cannot reproduce the original steady states. And to maintain the consistency about steady states, we need to add some correction term. Here, I call it rewiring. So you need to introduce some rewiring around the boundary you removed, and the boundaries of the parts you removed. And the precise formula is shown on the right here. So here, S22 is the stoichiometry matrix for the original system. So S22 denotes a stoichiometric matrix of this yellow part. But and to reproduce to keep the original steady state, we need to add this second term, the rewiring term here. Here, dagger denotes the pseudo inverse of S11. S11 is the part of the stoichiometric matrix of the removed part. Okay, and the derivation is actually relatively easy. So let me briefly sketch the derivation. So suppose that the system contains a buffering structure, and we are interested in the outside of the subnetwork gamma 1. So suppose that we are interested in gamma 2. Then the dynamics, we have two equations. One is for the subnetwork gamma 1. The other is for the subnetwork gamma 2. And because we are interested in gamma 2, we want to express things in gamma 1 in terms of gamma 2. So specifically here I expressed reaction rate in R1 in terms of reaction rates in gamma 2, like this. And of course we have there are several other terms. So from here to here, from left to right, I just solved this equation in terms of with respect in terms of R1. Okay, and then we substitute this expression into the second equation, and we obtain this complicated equation. And this can be done for any subnetwork, gamma 2 and gamma 1. But when gamma 1, the subnetwork gamma 1 is a buffering structure, we can prove that this term S21 times C is a kernel vector in S11. This term is 0. And about the last term, if we are interested in steady states, of course we can ignore this time derivative. Okay. So we can formally consider the equation by dropping these two terms. And this last this is a conclusion. So here so x2 dot is the dynamics of x2 dot x2, so that the concentration inside the gamma 2 is described by S22 times R2. This S22 is the original stoichiometry matrix for gamma 2 plus the correction term. Sorry, maybe I made a mistake. Yeah, so sorry. I need to multiply S21 on the here. So bracket inside the quantity inside bracket should be S22 minus S21 times S11 dagger S12. Yeah, sorry. And this equation should reproduce the steady state of the original system. And here we demonstrate this idea using a numerical simulation. And again, this is a network of central carbon metabolism. And suppose that we are interested in the green part of the network, the glycolysis, then we can ignore, for example, we can remove the yellow part, or we can further remove the TCAC, the blue part, and we finally obtain this simplified network. And here I demonstrate that these three networks are converge to the same steady state values. So here, x-axis is time, and after a long time, all of these three networks converge to the same levels. Okay, so in this way, if we are interested in a specific part of the network, then by using this technique, we can remove or simplify the network. And so we don't have to model the outside of the focal part. Okay. And so this is essentially the last slide. And finally, buffering structures can also be applied to study multi-stability. And yes, so and suppose that our system has multiple steady states. So, and in general, the multiplicity differs across the networks. I mean that some species may have two levels, but some other species may not show multiple states. So here is an example. So for example, in this system has three steady states, three stable steady states, and but the chemical species A only have two levels, and chemical species C has only one level. Only chemical species B have the three levels. Okay. So in this way, the multiplicity differs across species. So, and this raises a natural question. Which chemical species do we need to observe to distinguish or to identify the steady state the system occupies? So in this example, if the answer is B. So if we observe species B, we can determine which steady states the system occupies. Okay. And Yunjin Huang, a graduate student in our lab, provided an elegant answer to this problem. And the method is as follows. We first decompose the network based on buffering structures, and then compute the determinant sign of each subnetwork. And some determinant signs are positive definite, and some are negative definite, and some are sign indefinite. I mean, the sign cannot be determined from structured information, and the sign may depend on may depend on kinetics or concentrations. And then Yonjun Huang, he showed that observing species inside the sign indefinite subnetworks is sufficient to distinguish steady states. And yeah, we've posted this study on bioRxiv last week, so please take a look if you are interested in. Okay, here's a summary. So today I explained roles of buffering structures. First one is sensitivity, And today I didn't explain anything about bifurcation properties, but it can also, buffering structures is also useful to understand bifurcation properties of chemical reaction network. And buffering structure can also be useful to perform network reduction or distinguish steady states. Okay, so finally, I'd like to thank all of my collaborators, and thank you very much for your attention.